Hi everyone, my name is Ivayla Gyurova. I am a PhD student in Steve Waggoner's lab at Cincinnati Children's. And today I will talk to you about my project on the dynamics of adaptive natural killer cells in longitudinal analysis of CMV vaccine recipients. And before I start, I would like to take a brief moment to acknowledge everyone in the Waggoner lab for all of their support and all of our collaborators who have contributed immensely to the progress of this project. As well as I would like to thank the NIH for their generous funding. CMV is the topic of my research and it still poses a significant global threat. To give you a visual idea of the worldwide seroprevalence rates, we'd see that for countries with relatively lower known prevalence, such as the US, Canada, or some European countries, this prevalence is still considerably high, with rates above 30%. We see extremely high prevalence in some countries, as shown here in bright red. But what is even scarier is that for many of the developing countries in South America and Africa, we have no available data. Unfortunately, the lack of available data isn't due to a newly emerged viral epidemic. CMV is something that we have known about since the 50s. It was first discovered in 1956, but the first scientific publication didn't come out until 30 years later. And in 2000, we realized that CMV is also economically draining, with costs exceeding $2 billion in the US alone. And while we had some promising results from two different vaccine trials between 2009 and 2012, we still don't have an effective vaccine on the market today. 2 years from now, we're going to celebrate the big 65th birthday of CMV, and we still remain hopeful that we can send this virus to a nursing home. In the US alone, CMV is highly prevalent, with 50 to 80% of people becoming infected by age 40. And this comes to no surprise, as there are various routes of transmission, including vertical or mother to child, sexual transmission, or through transplantation. And while this isn't an issue for immunocompetent individuals, CMV poses a significant threat to babies born to the infection and also people who are immunocompromised, as it can lead to various complications, illness, and in many cases, death. Current vaccine efforts have only focused on the induction of memory T cell responses and antibody production by B cells. However, these approaches have conferred insufficient protection to CMV so far. So this highlights the importance of creating an effective vaccine against CMV in order to prevent the morbidity and mortality in immunocompromised individuals and allograft recipients, and also developmental disorders associated with congenital infection. A huge focus in our lab are these cool innate lymphocytes called natural killer cells or NK cells. And as their name suggests, they're good at killing viruses and they're also at the forefront of infection. In CMV specifically, they're critical for the control of CMV. Previous murine CMV studies have revealed that when mice are challenged with CMV, we see an expansion of a subset of natural killer cells marked by the Li49H activating receptor on their surface. And these NK cells, Li49H expressing NK cells, are responsible for clearing the infection, rendering mice resistant to CMV. While CMV establishes latency in humans, we see a similar phenomenon where when a person becomes CMV positive, we see an emergence of a subset of NK cells marked by an upregulated expression of NKG2C and CD57 on their surface. And in both mice and humans, we see persistence of these NK subsets, and therefore challenging that dogmatic view that NK cells are solely innate lymphocytes, and they can form long-lived populations of memory-like or adaptive-like cells that are superior responders upon reinfection. We had the unique ability to get samples from a CMV vaccine trial cohort, and this vaccine trial was conducted here in Cincinnati in the associated vaccine and trial evaluation units throughout the United States. And to tell you briefly about the trial, healthy adolescent females were given either 3 doses of placebo, which was sternosaline, or 3 doses of glycoprotein B subunit vaccine in MF59 adjuvant. And previous studies have shown that glycoprotein B is a potent inducer of neutralizing antibodies. So it served as a promising platform to the development of a successful vaccine. So the 3 doses were given at day 0, day 30, and then 180 of the vaccine trial. And the girls were monitored throughout the course of the trial. Blood was collected and PBMCs were saved and available for the purposes of the study. And I would just like to point out that these were longitudinal samples from CMV negative females. So the question that we asked here was, is this adjuvanted glycoprotein B vaccine sufficient to induce NKG2C? What we hypothesize here is that NKG2C expansion requires multiple triggers that are associated with HCMV infection, and therefore we didn't expect the increase of NKG2C following vaccination. And as expected, we saw no increase in NKG2C expression in both vaccine and placebo recipients. And the graphs depicted here represent one patient, one representative patient. Just to remind you, when a person becomes CMV positive, we see the emergence of these NK cells marked by the presence of NKG2C on their surface. However, recent evidence has also shown that another subset of NK cell emerges following infection. And these NK cells are marked by the loss of various intracellular adapter proteins of note FcR gamma, SIC, and E2. And what's more important is that these NK cell subsets have been shown to specifically expand in response to CMV antibodies. So going back to our vaccine trial cohort, we used these longitudinally collected PBMCs, and we again asked, is this vaccine formulation sufficient to induce this FcR gamma negative, SIC negative, and E2 negative NK cells? And what we hypothesize here is that these subsets would expand following antibody responses triggered by vaccination. And what was interesting here was that the answer wasn't as black and white as we expected. What we actually see are various patterns of expression. At first, we were excited to see expansion of these NK cell subsets in some of our vaccine recipients and 5 of the 20 tested vaccine recipients. However, later analysis of placebo samples also revealed that this expansion was evident in the same percentage of vaccine samples. Another pattern of expression we saw was a presence of these subsets at baseline and their subsequent decrease of expression over time. And again, we saw that, that subset of expression in both vaccine and placebo samples. But the most prevalent pattern was this ebb and flow pattern of expression throughout time, or this zigzag-like pattern that you see here. We saw that in over half of our vaccine samples and in half of our placebo samples. And also, if you noticed, from all of these patterns of expression, these intracellular subsets move together expression-wise. So how do we interpret these results? Just to revisit briefly, I would like to remind you that this is the first longitudinal study of NK cell subsets in CMV negative healthy individuals. All studies prior to that on NK cell subsets in CMV have only focused on the comparison of NK cells in CMV negative versus CMV positive individuals. So what we see in our vaccine trial participants was no change in NKG2C, which was consistent with CMV seronegative status. And the three intracellular marker NK subsets were marked with variation. We see expansion and contraction, and we also see that these subsets frequently move together. And none of these changes were linked to vaccine administration, as we also see these changes in our placebo subjects. So how do we explain these distinctions in our study? So we think that these differences are the product of the longitudinal nature of the analysis. And additionally, we think that it could be a product of the distinct pediatric nature or the underlying ethnic diversity of our study population. And we actually think that these subsets may be way more sensitive than previously thought, and they may respond to various environmental, inflammatory, or antibody cues. We also think that ebb and flow pattern of NK cell subset expression contributes to that CMV-associated preponderance of these subsets. So what we think is happening here is that when an individual is CMV negative, NK cell subsets oscillate but don't cross that threshold of subset detection. So they're not as readily detectable in CMV negative individuals. However, when a person becomes CMV positive, we think that what's happening is that these subsets have more rapid frequency of oscillations, or they're crossing that threshold more often, or they have an increased baseline or amplitude of detection, and that's why they're easier or more readily detectable in CMV positive individuals. So some of the remaining questions that we're also interested in pursuing here are: what causes subset composition to change? And can we modulate that with vaccine administration? And what function is mediated by these cell subsets? And with that, I would like to conclude my presentation and thank you all for your time and your attention. Greetings from Cincinnati.